Hello, my name is Marcus Horwitz, and I'm Distinguished Professor of Medicine and Microbiology, Immunology, and Molecular Genetics at UCLA, and former Chief of the Division of Infectious Diseases. I'm a member of the American Society for Clinical Investigation, Fellow of the American Association for the Advancement of Science, and a winner of the Oswald Avery Award for Outstanding Research in Infectious Diseases from the Infectious Diseases Society of America. My research focuses on the biology and immunology of a select group of bacterial pathogens called intracellular parasites. They have the unusual capacity to invade and multiply within human white blood cells, cells that normally protect us from infectious agents. These pathogens include the bacterium that causes tuberculosis, as well as bacteria that are classified as Tier 1 select agents because they are considered most likely to be used in a bioterrorist attack. These include the bacteria that cause tularemia, plague, and amyloidosis. Much of my lab's work has involved developing vaccines against these pathogens, and today I'm sharing about a novel vaccine we have developed against TB and a platform vaccine we have developed against Tier 1 select agents. Mycobacterium tuberculosis, the agent of TB, is the leading cause of death worldwide from a single infectious agent. It infects approximately 1/4 of the world's population, and each year causes active tuberculosis in approximately 10.4 million people and death in approximately 1.7 million. A vaccine called BCG was developed against TB over 100 years ago and has been administered to approximately 6 billion people, mostly in their infancy. However, BCG is only about 50% effective. Thus, an improved vaccine is needed, including a booster vaccine for people previously vaccinated with BCG. We have developed a vaccine that induces a strong protective immune response to the TB bacterium. Our vaccine, which uses an attenuated intracellular bacterium that mimics the life cycle of the TB bacillus to express 9 TB proteins, has been tested in 3 animal models and has demonstrated strong protection against the bacterium and a boost to previous immunization, making this vaccine not only suitable for unvaccinated people, but also as a booster for the billions of previously vaccinated people. With respect to Tier 1 select agents, we have developed a safe and potent vaccine platform to protect against the 4 major bacterial Tier 1 select agents, those of tularemia, plague, anthrax, and amyloidosis. Currently, a vaccine is available only against anthrax, and it is suboptimal. As with the TB vaccine, our platform vaccine utilizes an intracellular bacterium that mimics the life cycles of the target pathogens as a vector to express protective proteins of these target pathogens. In this case, the vector is a highly attenuated derivative of a tularemia vaccine previously administered to over 60 million people, and thus should be extremely safe. We have demonstrated that our Tier 1 vaccines protect mice who have been exposed to highly lethal doses of the bacteria that cause tularemia, anthrax, plague, and amyloidosis. Our tularemia vaccine has been independently evaluated by the Defense Threat Reduction Agency in a different animal model and demonstrated to be 100% protective against highly lethal doses of multiple strains of the tularemia bacterium. Of note, one of the Tier 1 select agents, the agent of amyloidosis, is a major public health problem independent of its designation as a Tier 1 select agent. Amyloidosis causes approximately 165,000 cases and 89,000 deaths annually in tropical and subtropical areas of the world, including Thailand, Malaysia, China, India, Singapore, and Australia, and there is no licensed vaccine against it. The U.S. government is interested in vaccines against Tier 1 select agents for administration to military personnel and first responders, and for stockpiling for potential administration to the entire U.S. population, should circumstances warrant this. The intellectual property for both the tuberculosis vaccine and the Tier 1 select agent vaccine platform is available for licensing. Thank you for your attention.